I'm going to talk to you today about mostly biological basis of resilience. And then maybe we'll talk a little bit, or mostly the day will be in the discussion will be from biological resilience to algorithmic resilience. And I found, thinking about this, I actually came across this quote from the World Health Organization from 1948. And it says, "Health is a state of complete physical, mental, and social well-being, and not merely the absence of disease or infirmity." And then there was a different, or an addendum to that in 1986, a clarification that mental health is a state of well-being in which an individual realizes his or her own abilities, can cope with the normal stresses of life, can work productively, and is able to make contributions to their community. And I thought that was interesting. And specifically, this is interesting because if I look from my perspective of being for 20, 30 years now in the biomedical research, the prevailing framework is to keep the research and to keep the clinical focus on understanding and preventing pathology. And we don't do much to understand the coping or the well-being or this health beyond the absence of disease. So this is really what we started with, thinking about the Berkeley Resilience Center that me and Shafi established and direct now, with the support of a very generous donor that stays anonymous. And there are three ways that we're asking this question. One is we're trying to look at biological data, and I'll focus on that today, and I'll show you that, and use and build and develop AI algorithms to harvest this multimodal data set and understand principles of biological resilience that probably we wouldn't understand otherwise. Secondly, we want to see whether we can actually learn from computer science, from AI theory, to inform the ideas of resilience paradigms. And thirdly, and maybe the most complicated one, is to try to take ideas that we learn from the brain and then use them to design, to architect better neural networks, to architect better AI and robustness. So I think you're going to hear about all of those three things in those 2 days that are coming. This is where I'm going to focus my talk this morning. So the vision that we started with was to uncover biological basis of resilience, how the brain adapts to stress, injury, aging, and using tools that will help us find things that we will not be able to do without. And then translate these insights into new strategies for mental health and neurological recovery. And also, as I said, use that to algorithmically resilient AI systems, right? So what I'm showing you today, and I'll give examples as well, is that I think it's not just uncovering something. It's not the absence of the stress response, but there are new ways that we're finding, new pathways that we didn't think are involved, but we're finding them just because we are focusing on the resilient individuals. And we want to bridge neuroscience and computational to reveal those shared principles, things like plasticity and adaptation and robustness, and advance the cross-domain understanding of how systems recover and thrive and evolve under challenge. So let's start with a story. Okay, this is some Berkeley Hills, or Sonol Park specifically, if you've been to the Bay Area. Very beautiful at this time of the year. This is last week us going there. And I want you to imagine that all of a sudden we're seeing a mountain lion. Okay, just imagine it in your body right now. You're seeing a mountain lion. There's Mike and an Alex that were walking there with us and saw the mountain lion. It sounds a little bit ridiculous, but actually it's not that ridiculous. Those are all sightings of mountain lions in those places. We had not met a mountain lion last week, but we did a rattlesnake, not that different. Okay, what is the first thing that happens? Your heart is pounding, you breathe a little bit quicker, your palms get sweaty. There's a response there, right? There's definitely a response. There's a stress response that comes with that. Okay, now, Mike and Alex froze there, and then they start making noises and trying to remember whether they're supposed to make noise or actually disappear or lie dead or whatever. But They get away from the situation. The mountain lion disappears. They go the other way. They go back to their home. They have a good supper. And a couple of weeks later, Alex is fine. He's back on the trails. He's running again. Mike, not so much. He's actually having nightmares, thinking about whether he should go back to running or not. Is it that trail? Maybe he shouldn't go on any other trail. Who knows if they're not able to sleep, not able to concentrate. Some different in the response there, right? So some different trajectory. And this is something that we all can relate to, right? Sometimes I give examples like, let's imagine ourselves in a terrorist attack. I decided not to do that to you today. So just think about some stressor and a group of people that are that come across that, and they're going to be a different trajectory in how we respond to that. Okay, so we know that there's a stress response. It's an acute stress response. It's what happened when they were on the trails, right? This response is vital, and it's necessary to survive, to adapt to challenges. In fact, in animals where we take away completely the stress response, the next time that somebody comes in to change their bedding, some of those animals will die. So it's a necessary response. And the way that the brain governs that is that it works like a prediction machine. It's actually there to predict threat. It's a threat detection machine, if we want to think about it in a machine kind of way. And from a survivability perspective of that, there's a reason for that, because the cost of false negative predictive error is much bigger than the false negative predictive error is much bigger than the false positive. So we want to be able to detect the threat. And now the question is, now the threat is not there, their life went on, what do you do with that? How do you compute the future threat and keep yourself both functioning in the world but safe? So there's a response. You may have heard it as the fight or flight or freeze response. How many times in my life have I said that? Okay, we could talk about memory as well and aging. The fight, flight, freeze response, which is a physiological reaction, occurs in response to actual or perceived harm. And that's important, right? So it's not just the actual mountain lion there, but it's also the perceived harm or thinking at home that there may be one there. What mediates that response is a whole host of biological responses, but there's a lot of important biological concepts that we can get from that. On the design of that, there's a very fast response, which is a nervous system response, so electronic signals that are going on, activation of two arms of the sympathetic nervous system. One would be for the threat and one would be for normal times. And there's a slower response that does something very similar, in which hormones are released. There's an axis of 3 hormones that are released from the pituitary, from the hypothalamus, and then the pituitary, and then the adrenal gland, and increase each other. There's positive and negative feedback responses, and there is a shutdown of the response. So all of those are the component of the system that come about to do this response that is vital to keep us running away from the mountain lion in this case, right? There are many, many ways that this system is actually plastic, because there are so many redundancies there, and there's so many checks there and balances on the response itself and where it could be. And also you could see that, for example, one of the hormones here, glucocorticoids, will have multiple points in which it inhibits its own production or the production of hormones on top of it, and also goes around and inhibits the electrical activity in some brain areas, and then increases electrical activity in other brain areas. So many, many points in which this could be fine-tuned. So it's a very plastic response that can be fine-tuned. There's, as I said, a lot of brain networks that are activated during the stress, different areas that we think about, and they do different things. So the prefrontal cortex is one of them, the hypothalamus, the amygdala is sort of the threat detector. This is the area that gets activated during stress. Prefrontal cortex executive functions over that. And then really we think about this today, not so much in brain areas, but in circuits that are connected and change how the whole brain is working. Every cell in our body has at least one receptor to the stress hormones. So it's a very orchestrated response. There isn't a cell that doesn't sense and respond to that. There's also an optimal amount of stress, right? You know that there's actually some amount of stress that is good for your performance. So if we look at stress here would be on the x-axis too little to too much, and the y-axis would be any kind of performance that you choose to put there. You see that some amount of stress actually increases your performance and you do better. That's a positive stress. What do we know about this? It's short term, it's perceived within our coping abilities, it motivates and it focuses energy, it improves our performance, and it can protect from future stressors. So it actually might be sort of a immunization against future stressors. But then there's the amount where it's too much and we see a decrease in performance in a lot of different ways that we measure performance. So the very first thing that I get, the question that I get when I teach this in class or when I talk about it in, when I'm interviewed about stress, which happens a lot, is, well, what is the right amount? I need to know what is the right amount. And there is no such answer for that. There is no right amount that I can tell an audience, because each one of you will have a different right amount. There's individual variation in the stress response, and you can imagine that you would be one of those people that actually do really well with less amount of stress. In the medium amount is already too much for you, or you're one of those people that really need the drive to get this push up, right? When we think about those trajectories of vulnerability or resilience, we can imagine now this response of an individual and see that if we put some line on psychopathology, or, this could be, let's imagine here, depression, PTSD, some other generalized anxiety, there will be the green individual that takes a small hit with the stress or trauma exposure and they're fine. There's the yellow individual that actually dips into meeting the criteria to be diagnosed as one of those psychopathologies, but very quickly recovers. And there's the red individual that takes a hit and stays there. And you might imagine our Alex and Mike from before, where Alex was somewhere the green or the yellow and Mike was a red one. So let's talk about this individual variation for a minute. Around, pretty shocking thing here, around 70% of individuals in the US are exposed to at least one criterion A traumatic event in their lifetime, something like threatened death, severe injury, sexual violence. A substantial portion of individuals will develop symptoms in the weeks or maybe the month following the trauma, but most will recover within the first 3-month post-trauma, and approximately 10 to 20% of exposed individual will develop a persistent adverse post-traumatic sequel. So PTSD, anxiety, depression, addiction, compulsion, something like that will stay with about 10 to 20%. And we want to understand those 10 and 20%. And in fact, what I did for the last 30 years of my research was understanding the progression towards those pathologies. So, and me and many, many others, so the field now knows that there's a dysregulation of this HPA axis, this hormonal axis. There's heightened amygdala activation, that threat reactivity is heightened in post-traumatic stress disorder. There's a weakened connectivity of the prefrontal cortex over the amygdala, so the area that tells us it's okay. This was last week. There isn't a lion. There's not likely a mountain lion on the trail this week. There are things like fear generalization or impaired fear extinction, meaning when I go back and back again to the same place, I need to relearn that context as safe, and that does not happen in PTSD. Or generalization, it happened in that area, it will not necessarily happen in another area where I go to. No, now I generalize my fears. And there's memory fragmentation, so PTSD is very much of a memory problem. There's also shift in pathways that are active. There is much more rumination. There is much more internal thoughts. There's much more threat reactivity, and there's a lot less of the top-down regulation of thought, actions, emotions, salience, and executive functions. Right? So, big differences. Why? Why are those differences? You all sit here, take a second and think, what is the stress, the governing stress factor for you? What do you think in your own life, biology, experiences, meditation that you did this morning, whatever it was, what is something that works for you or that changes your own stress response, right? And there's a lot of, I was going to ask you, but for time's sake, you'll stay with the response for yourself. That's worth it as well. And we'll say that there are things like genetics. There's things like life history. What were we exposed to when we were kids? There's epigenetics. Sometimes even in the field, we actually have newer data to talk about transgenerational effects, what happened to our grandparents. Personality, things like optimisms seem to be something that changes the stress response. Social, cultural, nature of the trauma itself, how severe it is it a collective trauma? Is there a social network of support? Do we have a sense of control over it, controllability? How did we sleep the night before that happened? What is the physical activity level that we had before and after? All of those things change. Now, I study mice mostly, so genetics is not really a question there because in the mice that we study, they're mostly genetically available. So we don't go to differences in their genes unless we are the ones that are changing their genes. We think of them as identical. Life history, we can control for that. And they all grew up in the same animal facility and had the same life. Personality is a question. I don't know. I worked with them for many years. I'm still to be convinced, but there's definitely people there that talk about a personality of a mouse. All the things like cultural and controllability and severity are either not there or we can control for them, meaning we can focus on the biological factors, on neural activation, glial activation, hormonal activation, vascular, immune, metabolic, and circadian regulation. Those are things that we can measure and we can focus on and see, are there differences? And even in those genetically identical mice with the same life, we get a big difference of responses. So the approach that we take to asking this question in my lab, and this is the lab right now, is the multimodal approach. We use a lot of different techniques from molecular, cellular, physiological, behavioral, system, and computational tools. We look across brain areas and systems. We look across multiple cell types, neuron, glia, endothelial cells. We look across systems, vascular, immune, endocrine, metabolic, across the lifespan. There are different responses in adolescence than in aging, for example. And we look across diagnostic borders. So we don't pay attention so much to neurology, psychiatry, psychology, or even within psychiatry. Is it anxiety? Is it PTSD? Cross-diagnostic. Let's look at the effects of the symptoms. And this is the pipeline that we use. So we have mice. Importantly, we take male and female. It sounds obvious. I will tell you it's not obvious in the field. So we take those mice and we expose them to some adverse event. Then we go through deep behavioral phenotyping. I'll say something about each of those in a minute. We go through the behavioral phenotyping to decide some resilience, either cutoff or a continuous measure. And then we take a lot of other measures of those animals. We'll see those in a minute. We harvest the tissue afterwards. We look at blood-brain barrier markers. We look at immunohistochemistry, and we do omics, transcriptomics and proteomics on those animals. Then comes in all those AI-guided analysis to propose candidate mechanisms of resilience. Hopefully synthetic data will also come in there. And with that, we then go into modeling candidate mechanisms. The modeling that we do is with transgenic animals, so where we change the genetics of them. We have cells, human cells that we grow in culture, or human brain organoids that are cells that come to a 3D model of the brain to then identify and test drug and genetic therapeutic candidates. Okay, so that's the pipeline that we're going through. How do we stress a mouse? Very similar to the exposure that we saw before. Here we expose them to their own predator, which is a fox urine, or we do a chronic unpredictable stress. So every day something else happens. One day you're shaken, one day your bedding is wet, one day you get an electrical shock. Social defeat stress, another paradigm where it's sort of a bully paradigm. There's a bully mouse in your cage for 10 days that stresses you. We can socially isolate or other things. We also look at physical stressors. traumatic brain injury and concussion. We have a model that we developed of a modified weight drop, closed head injury. So very similar to what would happen to, let's say, a football player that get hit multiple times during a season. It's not necessarily even a concussion, but could be a subconcussion. I'll show you some data from that. And we look at aging and neurodegeneration as another model for that. We have mice that are just naturally aging, and I'll show you data from them. And we have mice that have a human gene that mimics Alzheimer's disease. So one way is we do those models, and now we're going to look at what are the animals that are vulnerable or resilient. Another one is we look at selective vulnerability within brain areas. Let's look at why one brain area shows a damage and the other one does not within an individual. Okay, how is the deep behavioral phenotyping going? We're looking at a lot of different ways to measure things like avoidance or fear, depression, social behavior, compulsivity, learning and memory, and motor function. We can talk about how each of those are done, but I'm looking at the time, and if you want, you'll ask me about it. And then we get some kind of a composite measure in a lot of different domains. And this is important to us, right? So we're not just looking at one assay. And in that composite measure, we can find animals that are susceptible, and we can find those blue animals here below, the blue animals here below that are resilient, that look not different from the control population, statistically not different from the control population. There's a lot more that one can do. So there's a whole conversation to be had about behavioral in mouse, and it's not the best way to do things. Even when you do this deep behavioral phenotyping, there's probably much better ways and dimensions that we can interrogate beyond those traditional assays and what they capture. For example, we can only do those assays once in a mouse. Once they've seen the maze, you can't test them again. So we're trying to put in things that are more complicated. For example, this is a collaboration with Jorge Palop at UCSF and those students in my lab that are looking at VAME, variational embedding of animal motion. You're looking at the motion and just trying to get a machine learning paradigm that extracts data and tells us about whether there's any differences. We are agnostic to what the differences might be. Jorge had just published this beautiful paper in Cell Reports showing differences in learning in Alzheimer's disease mice. We're trying to do that on stress and seeing whether we get that. And we see this inter-individual variation across all models. So when we have models of stress, up top you see immobilization and predator odor here. So the mice is immobilized and they're smelling the fox urine, they can't run away. The social defeat paradigm and other stress paradigms, we will get susceptible and resilient individuals to that. When we do the social defeat, for example, we see that we get different groups. There are groups that have a social effect, there are groups that have an anxiety effect, there are groups that have a compulsivity effect. They're not necessarily the same group, or they're not the same group. So that's an interesting idea, is we could talk about resilience, but domain-specific resilience. Then repeated mild TBI. Those are the animals that get 4 hits, very, very mild hits. We don't see any differences in their neurological scores after 1 hit, but after 4 impacts, we start seeing an effect in a susceptible group, not in the resilient group. So there are animals that get the 4 hits and they're just fine, animals that get the 4 hits and we see a big decrease in their neurological scoring. And the same goes with aging. And we can run statistical models on that in GMM clustering and see that they cluster really into 2 populations. The population that ages very nicely and seems indistinguishable from the young ones. This is by neural activity, paroxysmal slow wave events. And there's an other group that ages but have thousands of those paroxysmal slow wave events per day. Those are events where we can think about our brain sort of slowing for a second there. Okay, so we have this resilience spreading right now. We did it on a group of animals and we get all of those different ones and we can compare them. What do we compare? What are the measurements that we take? We do telemetry where we record neural activity, or we record from the brain, or we record from the body temperature and activity that tells us about circadian rhythm as well. we also take a miniature microscope and implant them into their brain, and we can record activity in that. So this is an example in this movie. You see recordings of calcium imaging, calcium activity. In this case, we wanted to look at the calcium activity not in the regular cells, not in neurons, but in oligodendrocytes. And this is what we came about. And we can do serum analysis and tissue collection for the omics. And what we get here is a really interesting finding. What we found initially is that the biggest response is in the resilience group, right? So it's not a lack of a stress response. This group has a very active, big response that drives the differences in resilience, right? So we can find a lot of unknown targets there. And the mechanisms for that are neural and glial and vascular, and all of those. I'm going to tell you about very, very quickly about two of them. So the first vignette is about myelin plasticity. It's a big story with papers going back several decades from our lab that deals with the myelin. The myelin is the sheath that wraps around connections between neurons and was thought about as something that is pretty stable in the brain. But in the last few years, we realized, us and many, many others, that there's adaptive myelination, that myelin is actually a plastic thing that changes throughout the life, and it changes with response to learning, to motor function, to a recent paper from the Monge lab, opioid addiction, and we found that it changes with stress. We also know that difference in myelin organization occurs across multiple psychopathologies, and it's a potential transdiagnostic risk factor. And if myelin changes, it will change conduction timing, synchronization between circuits. It limits the connection between cells. So it's critical for neural circuit precision, and dysregulation of it may contribute to psychopathology. I'm jumping over all of the mechanism to just tell you that at the end we found that myelin changes both in rats and in humans in a very similar way, where you see changes in myelin and they correlate with CAPS score, with a PTSD score, or they correlate with the anxiety score of those animals. And in fact, not in the way that we would predict. The more myelin that you have in gray matter in the hippocampus causes, for example, greater anxiety. But it's region dependent. So in other brain areas, there are other changes that we could have seen. We also know that we can manipulate that. We can take a viral vector that drives myelination in that area. And when we do that, we get animals that develop anxiety without exposure to stress. And lastly, from this unpublished data coming from Tom Nalen in UCSF and Jenny Stevens in Emory, literally weeks ago, we show that this resilience is associated with the myelin content in the human brain as well. So the resilient individuals have less of that myelin overshoot in response to trauma that they were exposed to, and in interesting areas of the brainstem, and the reward circuitry. And that maps very nicely on the functional effect, in which there's greater engagement of the salience network in response to reward cues, and lower engagement in DMN in response to threat and reward stimuli in resilient individuals. A really interesting part of that is that we recently started looking at psychedelics. If you're in neuroscience and if you're studying PTSD, you have to study psychedelics. And psychedelics work on a receptor that is expressed in neurons, and therefore we're only studied in neurons. But turns out that this receptor is also expressed in the cells that produce myelin, those oligodendrocytes. And when we look at that, we see that those cells start to have a calcium increase. They synchronize those oligodendrocytes and they produce more myelin. And if we take away this myelin producing activity from them, we genetically knock it out, we show that those animals don't respond to the psychedelics in the same way. They don't have this anxiolytic effect. Vignette number 2 is the blood-brain barrier. I will do that very quickly. Again, there's a lot of literature from our lab to study that for many years. So I'm not going to tell you the details of that, but I will just say that the blood-brain barrier itself is a protective shield at the border of all of the blood vessels. So the cerebral vasculature has a different lining to it that filters things differently and protects the brain, keeps it as a privileged environment. Things from the blood don't cross and don't get into the brain. Cells or proteins don't cross very easily. We see dysfunction of this in a lot of different neurological and psychiatric disorders, and the symptoms could be correlated with the extent and the location of blood-brain barrier. So we've been studying it for many, many years together with Alon Friedman at Dalhousie University. And what we have focused on is a blood-brain barrier breakdown in traumatic brain injury. Those are American football players that were very young people, that about a third of them show some dysfunction of blood-brain barrier at the end of the season, even without a concussion. And we look at this mouse model that I told you we developed to look at, it's a very similar head rotation to what happens in football. Here, you remember we looked at resilient animals and susceptible animals by neurological scores, and the susceptible animals are the ones that have blood-brain barrier dysfunction. Aging, very similarly, when at the top you see humans at different ages, at the bottom you see mouse at different ages, and with age we see an increase in blood-brain barrier dysfunction. In fact, over the age of 70, about 60% of the individuals will show some effect of blood-brain barrier dysfunction. There's a sex bias to it, but we'll talk about it now. And then we spent many years understanding this mechanism. And the mechanism involves a blood protein called albumin that gets uptaken into the brain because the blood-brain barrier doesn't work as well. It gets uptaken into cells that are called astrocytes, binds a receptor there, a neuroinflammatory receptor, changes dramatically neuroinflammation in the brain, then causes a pathological signaling cascade and ends up in neural dysfunction and cognitive impairment. When we look at the neural dysfunction, we see that we can find it in the aged individuals that are the susceptible one, but not in the resilient ones. In the TBI, the concussion mouse model, we find it in the susceptible ones, but not the resilient ones. And we also see it in Alzheimer's disease model mice or patients. This is all correlation, it's not causation. So we did causation by two ways. One is a gain of function, where we take the protein, this albumin protein, and through a pump, put it into the brain and ask, is that enough to cause aging of a healthy young age otherwise, or by loss of function? So gain of function, yes, it's enough. 1 week of this protein being delivered into the brain causes a change in gene expression signature, in synaptic transmission, in inflammation, in senescence of astrocytes, and in neural dysfunction and cognitive dysfunction, so it looks like an old brain. By loss of function, we did it by two ways. One is we created a transgenic animal in which we knocked out this receptor for this protein out of the specific cell type, and that was enough to block the response. And we created a small molecule drug that blocks specifically the phosphorylation of this receptor. The receptor is TGF-beta receptor of the ALK5 kind. And when we block specifically the phosphorylation of this one, we restore blood-brain barrier function. The blood-brain barrier comes back to what it would look like in a young animal. And in an old animal, it reverses inflammation, it blocks astrocytic senescence, it restores the astrocytic function, it reverses the neural dysfunction, and it restores the cognitive function. So you can see here how quickly it works. This is in fact within a day. Those animals are recorded with the ECOG, and then they get, and you see the red ones are the susceptible ones, the resilient ones are the orange, and within 1 day they mimic the resilient ones. We block the blood-brain barrier, we block the neural function, we improve memory, and now they learn as if they were a young mouse, and they show the biochemistry and markers of a young mouse. Okay, so those were two vignettes. There's a lot more on that if you want to ask me in any of the breaks. So to summarize, we center the research on resilience to uncover novel mechanisms and therapeutic targets. We use AI-driven multimodal pipelines to map the molecular, the circuit, the systemic signatures of resilience. And I gave you a proof of concept with 2 different stories where targeted modulation of gray matter myelination, or BBB integrity, actively engineered resilience into that system. I have the current open questions, but I might say, we have time? Okay. So current open questions, and where are we going to from here? We want to think about resilience as a static trait or dynamic process, as a state transition, and we think it's a dynamic process. We know from human literature for sure. It's a little bit harder to ask this question in mouse because we can only look at one time point. So we're starting to think about ways in which we can look across times and also learn from the human about this dynamic process. What do I mean by that? For example, when I describe those individuals that went through trauma, went to the ER, then went into the Aurora study that Tom and Jenny were doing, those individuals, some of them show resilience at 2 weeks, some of them show resilience at 6 months. So there's the static resilience at 2 weeks, there's the dynamic resilience that happened for some of them, improving over the 6 months, and there's one that don't result. So we need to take into consideration those things. We also want to start implementing all of those data together, this multimodal data. So it's a lot, right? When I say we do omics, we have hundreds of thousands of hits there. When I say we do behavioral, those are many. So all of those omics data need to come together and be modeled in some way that represents that whole state of the animal. And we're trying to not just focus on the brain, but also have this idea where we're looking at things like their circadian rhythms, their metabolism, their immune function, all those peripheral things that we don't necessarily immediately think about when we think about the brain. All of them need to come together, and creating some kind of a mathematical framework of resilience or modeling ways that incorporate all of that. We've been talking and with Kostas and Yanis, and you'll hear about it, about their work later, and maybe you can help us make the connections here, but there's a potential for synthetic data or generative models for testing hypothesis. At the very bottom, at the very first level of entry into this is we just don't have enough mice. When we do it, we're limited with the amount of mice that we're doing, but now we get that the variability is so much greater than when we think, and the variability is greater in each one of those domains. So you can imagine that really what we need is a much bigger pool of data, and the synthetic data could be where we use that, and then bring it back to human translation and precision medicine, taking into consideration another level that we don't look at, which is the genetic data. And then how do we translate back, right? So from the biological resilience, what can we learn for algorithmic resistance? This is something that I think we'll talk about in the panel later on. Right? The lab members, you saw them along the way, the collaborators, and the funding that allowed that. Thanks. Yeah, I think I have 3 minutes that you started late for questions. Oh.
This is so fascinating. Thank you very much. I have a question related to attention, a word that in the machine learning community is always important, but it seems to me here that, or can you describe how sort of attention changes in these stress contexts, like either the myelin, I guess I learned the word, right? Is it a word? Or other mechanisms that sort of change what the attention is and therefore change then the processing or the resilient, or does it have less to do with attention?
No, it has a lot to do with attention. So the brain circuitry that I glanced over very quickly are ones that the ones that help with focus and attention and computational powers and so on. Those are the ones that weaken during a stress. So during acute stress for sure, and then there are plastic changes in some individuals over time. So those are ones that if you look at individuals with post-traumatic stress disorder, they might have weaker attention mechanisms. Yeah, thanks.
Thanks, that was a great talk. One question, I'm wondering where you are in the ability to predict. We had these nice decomposition into two kinds of animals, ones that were resistant, ones that were susceptible. To what extent can you build predictive models of which animals are going to be in which bin before the stress is induced?
Yes, so this is exactly
where the synthetic data comes in.
Something that we want to go to, where the synthetic data is going, but also where the modeling, I think, is going, is that we should get at the end. And I just want to say it's not, bins is one way to look at it. I actually, I didn't say how we do those bins or what are the statistical models that we use for that. There's a cutoff model, there's a z-score, there's different ways. But there's also probably other ways to look at it that we try now, which are continuous data, right? So you're not necessarily just resilient or susceptible. You could be anywhere on that spectrum, and that those correlations seems to hold on very nicely with some of the biological things. Now, can we get at the end to the point where I get all the data, I give it to you, and you come back with a model that says, I predict that this and this animals are the ones that will behave this way, and I can tell you, oh, okay, let me pull out my behavior data and see. That's exactly where we want to get to it. We were talking about it with Shafi yesterday, but that's the idea, that we'll get there. Yeah, so it's predictive, and then also understanding the mechanism helps us, and we find newer ways to, yeah.
Kudos on optimal cognitive stress that you achieved during your session. In terms of...
I would say I was on my personal stresometer.
So mice, humans, engineering resilience. Is it possible you could intuit or conjecture if a human reconditions ourselves through martial arts training or other kinds of mental stress training, are we able to move on that continuum from red to green?
Yeah, I'm absolutely sure it is. I myself practice meditation for many years and mindfulness, and I teach that, and it's because I believe that's the case. I can give you, okay, here's a very obvious thing. In the past, if I had to give a public talk, I would be close to fainting. I hate talking in front of people. Now, we can talk about whether I do it well or not, but I can do it now. So clearly, we change with training, right? And with practice and with all of those things. It's a lot of plasticity. So I'm sure there are a lot of different ways. One of the obvious ways in mice is physical activity. A stress outcome is very, very different for animals that have a running wheel in their little dorm room or not. It's better if they choose to go on it. It's not better if you make them. But if they're the one choosing, then yes, then the stress response is a lot less. They look more resilient. Yeah. So there's a lot of ways to do that. It's not going to be just pharmacology for sure. Yeah. Or
tough act to follow. Thank you.
So you mentioned trying to engineer resilience. Can you envision any possible second order effects of trying to engineer resilience, like effects on other attributes of those subjects if you were to engineer resilience?
Yeah. And it actually, I want to say, maybe, I don't know if that's what you meant, but it's not 100% clear to me that in all cases, engineering resilience is the right thing to do, right? There are times where not being resilient is maybe the better response. If you think about a mouse, they respond to what the environment is. And there's programming. There's transgenerational programming, and there's programming in utero, and there's programming throughout their life to stress cues that come from the outside. Meaning, if they're a very anxious mouse, it's probably because the environment is not safe. And they're being very hesitant to explore new places might be the thing that actually protects them, right? Now, in human terms, we always think about performance. And we want the performance to be, but the performance is in context of the environment, and the environment is not an environment that allows that. And even if you go to the hormone level, we used to think about stress response. What you want is a smaller stress response, and that's not true. It is now very clear that what you want is a large stress response at the right time, being shut off at the right time, because the lack of a stress response, the hypo-responsiveness, is a predictor of PTSD. Right? So it's a lot more complicated than, let me just, I have the secret to a resilient individual. It's always in context. Is it what you meant with your question?
Yeah, thank you.
You said the hypo response is a predictor of
PTSD. Yeah, hyper responsiveness is PTSD.
Okay.
Thank you.